Okay, good morning. I think two more people and everybody will be in. So it's Friday, it's 8:30. So Vladimir, what do we do?
Talk about vision research.
We're celebrating vision research, and today we have really outstanding scientist, Jessica Morgan. As you know, imaging really made a major impact on ophthalmology, clinical practice, as well as on research. And so we will have Jessica today telling us about her progress in novel way of imaging the retina. With that, let's just go to Francis for introduction.
Good morning, everyone. My name is Francis Aguas. I'm a first-year PhD student here in Dr. Andrew Brown's lab at the UCI CTVR. It's my pleasure to introduce today's distinguished speaker, Dr. Jessica Morgan. Dr. Morgan is an associate professor of ophthalmology at the University of Pennsylvania and director of the Center of Advanced Retinal and Ocular Therapeutics. She holds undergraduate degrees in both physics and mathematics and obtained her PhD in optical engineering at the University of Rochester, where she completed postdoctoral research with Dr. Jean Bennett and Dr. Edward Pugh Jr. Dr. Morgan's research focuses on adaptive optics with scanning light ophthalmoscopy, or AOSLO, to non-invasively image the structure and function of individual photoreceptors in the living human retina. Her lab leverages AOSLO to study photoreceptor phenotype and degeneration mechanisms associated with inherited retinal diseases such as choroideremia. Additionally, the Morgan Lab incorporates functional testing protocols with AOSLO to study photoreceptor function at higher resolutions, with the goal of developing functional biomarkers indicative of retinal disease pathogenesis. Dr. Morgan's work is well supported by multiple NEI R01 grants and the Foundation Fighting Blindness. Of note, Dr. Morgan was selected to participate in the ARVO Women's Leadership Program, and several of her students have been recognized with Young Investigator Awards, a testament to her mentorship prowess. She has published numerous manuscripts and abstracts, serves as reviewing editor for several journals, and holds patents for methods of imaging the eye and assessing photoreceptor function. With that, please join me in welcoming Dr. Jessica Morgan for her talk titled Adaptive Optics Ophthalmoscopy for Understanding Photoreceptor Structure, Function, and Treatment Response in Choroideremia.
Well, thank you so much, Francis. That was quite an introduction. I am really honored to be here today. Thank you, Chris, also, and Grazina, thank you very much for the invitation. It's great to see so many people both in person gathering for this and also online. I'm really looking forward to our time together. Let me share my screen here. And I think that looks great. Okay, so right as you just heard, my lab specializes in advanced imaging techniques, in particular adaptive optics, to study the structure and function of the retina. And so I'm going to go ahead and jump right in here. I want to thank our funding sources. You just heard about a lot of them. The work I'll be presenting today really is rising from our NEI grants primarily. And then I really, before I dive into the science, I want for sure to acknowledge the people that make it happen. So all of my current and former lab members who have done such wonderful work that I'm going to tell you about today. In particular, I'll be talking about my graduate student work, Angela Zhu, postdoc, Ray Warner, my lab coordinator and manager, Yu Jiang, as well as work that came from Rob Cooper and Will Tooten's time as postdocs in the lab. All right, so with that, I want to jump right in. As you just heard, my lab specializes in adaptive optics imaging with the goal of looking at the structure and function of individual photoreceptors in the retina, in the living retina. And so this is a technique where we can measure the optical aberrations of the eye, and through use of a wavefront sensor and an adaptive mirror or deformable mirror, compensate for those aberrations in a manner that lets us take a high resolution retinal image. And so here is an example of a fundus photograph that is taken routinely in ophthalmology clinical practices, just as really a standard part of their visit. But with our adaptive optic system, we can effectively zoom in to the extent that we're able to get images that have high enough resolution that we can see individual cells. And so here is an example image from really the central fovea region, where each individual spot in the image is a single cone photoreceptor. And in that foveal region, the cones are packed in. They're small, they're tightly packed, and that leads to our high spatial vision, high acuity. If you move into the parafovea, you see the cones get a little bit larger, but they're still really regularly arranged in a tight hexagonal packed mosaic. And then as we move out a little further, really still in the macula, but in, I guess, really the perifovea region, you see the cones become even larger. They now appear as a bright spot surrounded kind of by a dark ring. And then the rod mosaic starts to infiltrate in between the cones. So each bright small dot now is an individual rod photoreceptor. And so these are example images of what an adaptive optics montage can provide in terms of looking at the structure of the photoreceptor mosaic. Today's talk, I'm really interested in the science and the application of our imaging technology. And so I want to guise all of the methods that we use into the study of retinal disease. And I'm going to do that by talking about one particular inherited retinal degeneration called choroideremia. So just permit me a short minute here to talk about just what's known about choroideremia, inherited retinal degenerations. So first, choroideremia is X-linked. It is rare. And as an X-linked degeneration, males are symptomatic and females are carriers. The degeneration is caused by mutations in the CHM gene, which encodes Rab escort protein 1. And loss of this functional protein results primarily in loss of the photoreceptors RPE and choroid, despite the fact that the protein is ubiquitously expressed throughout the body. So when we look at a fundus photograph of someone with choroideremia, this is a typical image that you might find. The patient has atrophy starting in like kind of the mid-periphery that is ever encroaching in towards the central foveal region. And so this island of what is called retained retina is located centrally, and it is actually the dark part of this picture, with the mid-peripheral atrophy showing up as the bright regions of this fundus photo. And in choroideremia, you'll hear people talk about what's like typical scalloped edges, which is essentially this border of degeneration that's ever progressing inward. So patients present with loss of their night vision, primarily first, as well as a reduction or constriction of the visual field. And then finally, later in the disease process, loss of acuity as that border encroaches into the fovea. So we have taken a number of images now using our adaptive optics system in patients with choroideremia. And here's an example now of the adaptive optics image that you would find over this central macular region of the retained retinal area. And so that is seen here now as the larger adaptive optics image. And within that central fovea region, the cone mosaic is present. You have this kind of loop that shows up in their area of retained retina, that's this encroaching border right here that kind of dips down within our mosaic. But then the retained island is back. It has kind of this moon shape for this person. And you can see the photoreceptor mosaic again as you move out across that temporal parafoveal region. So if I just zoom in, I can show you some structural images of the photoreceptor mosaic within this region. This is, again, that central foveal region. You can see the cones are tightly packed in within this retained mosaic. I've identified individual cones in the top left corner of this image, just to give you an understanding that if we see the cones, we can identify them and use those identifications to quantify the image that we get into metrics that can come out from these images. If we look a little bit further, again, we can see the photoreceptor mosaic remains present up until that edge of the atrophy, the border of the atrophy, which is very quite sharp in its demarcation of where the photoreceptors are present here on the right side of the image, as opposed to where the photoreceptors mosaic has been lost or is no longer visualized in that top left corner. And then if we continue to look at other structural features of this particular individual, when we move to the outside edge of the border there, we're able to see what's called an outer retinal tubulation. And this is when you look on phos, which is like down onto the retina, an outer retinal tubulation has this finger-like protrusion appearance where you get a kind of a long spindle that comes out from the mosaic. And we know from OCT that this is actually the retina kind of folding in on itself and very late stage disease in that region. So we want, as I said, to be able to take these pictures now and quantify metrics that can come from them. And so in the most simplest version of quantification, we can take the image and essentially just count the cells. And so one of my graduate students has worked on this, where we've looked at regions of interest from control eyes in comparison to choroideremia eyes. We identify all of the cones in those regions across the groups, and then can come up with measures of cone density. And what we find is that even within that centrally retained area, where our patients many times maintain their high visual acuity, 20/20 vision, they are still exhibiting reduced cone densities in those areas, in those central areas. Beyond looking at cone density, we can also look at the morphology of cells. And so here now the student, in addition to identifying the cone centers, has also segmented the cell borders. And from a qualitative standpoint, you can look and say that first, yes, you see the cones are less densely packed in choroideremia, but you also note that they qualitatively appear larger. And so we quantified this by looking at cell area in choroideremia, and again found that the cell area is higher in choroideremia in comparison to control. And so these are ways that we can identify not just the full mosaic parameters, but also really look at individual cone morphology. And it's still work in progress to understand whether this enlargement of the cells that we see is the result of the cones having more space and therefore expanding kind of to fill that space, or whether this enlargement is really part of the degenerative process where the photoreceptor is on a path towards degeneration.
Jessica, all those patients are genotyped?
They are, yes. So these are all confirmed choroideremia genotype patients.
I don't know much about genetics of choroideremia. Is it one mutation or is it a spectrum of mutations?
It's so it's a spectrum of mutations that all affect the CHM gene. And those mutations result in a lack of functional Rab escort protein 1. Okay, so I want to transition here and talk a little bit now about cone function. And what I'll start with impressing upon you is the idea that just because we see a cell is present in our image, it doesn't necessarily mean that that cone or that cell is functioning or contributing to vision. And so we really want to be able to understand, are the cells in the mosaic all functioning the way they should? Is there reduced function? How can we tease out additional information beyond just the cell morphology? And so my lab is currently involved in 2 techniques to do that. The first is adaptive optics microperimetry, and the second is a up-and-coming technique called optoretinography. And so I'm going to talk briefly about each of these 2 functional measurements that we're able to do still through our adaptive optics imaging capabilities. So first, for microperimetry. Microperimetry is a technique, it's used in the clinic without the adaptive optics component, but essentially you image the retina at the same time that you present a visible stimulus. And that the retina is tracked so that you know where the visible stimulus fell on the retina. And then you ask the person, did you see the flash? Did you see the stimulus? And they push a button, in our case, a yes, no response. Yes, I saw it. No, I didn't. If the person says, yes, I saw it, then we present another stimulus in the same location, but with a dimmer light or a lower contrast, and ask the person again, yes or no. If the person says no, then we present a brighter or higher contrast stimuli. And in this way, we can effectively staircase in on what the threshold level of vision is for that particular retinal area. And that's the same way that it works in the clinic. But with the adaptive optics, we then have the higher spatial resolution. So this image that I'm showing here is really meant to illustrate the type of stimuli that are able to be presented. So here's an image of the choroideremia subject that we have. The border of atrophy is right here. So we're right at that border location. And this yellow spot that's placed on top of the image is the size of what's called a Goldman 3. That is the size of a stimuli that is typically used in conventional or clinical microperimetry. And as you see, the size of that stimuli placed on the border would encompass, it's larger than perhaps the region over which there's a transition in structure. And so with our adaptive optics, we're able not just to image the cells, but also to confine our stimuli to a smaller region. And so here now is that adaptive optics microperimetry spot that we were able to test in this particular patient. And what I'm showing is the true size of the stimulus that was used. So in this case, we did stimulate, about a dozen cones altogether. But with this adaptive optics microperimetry technique, it has been demonstrated that you can stimulate down at the level of a single cone photoreceptor. So for this particular location, we undertook our staircase algorithm of testing and found that the patient was able to see the stimuli presented in this region, and we could measure a threshold sensitivity for that group of photoreceptor cells. We then tested in the region that is beyond the border, beyond the atrophic border. And in this case, no matter how bright or how high of a contrast we presented, the maximum contrast that our system was able to present, the patient was never able to see the spot. It just didn't, that area of their retina is not contributing to their vision. And that, makes sense because we believe from our images that the photoreceptors are no longer present in this retinal area. So then the question became, well, what about essentially the transition region? And so when we test right at that border, again, we find that there is sensitivity to that stimuli, though it is a bit reduced from the photoreceptors that are really very closely adjacent. And so this leads to several important findings related to structure function correlation. So in choroideremia, the structure and function of the photoreceptor mosaic is very highly correlated. They're very closely related with a very sharp transition zone between where those photoreceptors are functioning and not. And that then also provides important evidence for how to design or how you might think about designing outcome measures for clinical trials. And in a case like choroideremia, I would suggest that evidence that so tightly correlates structure with function would enable an understanding that a structural endpoint for a choroideremia trial might be completely acceptable for understanding the safety and efficacy of the therapeutics that you're delivering. So that is, one bit of information that we were able to glean from this microperimetry testing. Yeah, go ahead.
May I have a few questions? First, I wanted to ask about the size of the stimulus.
Sure.
The size of the stimulus and why this particular size?
Right. Okay, great question. So we picked a size that was close to commensurate with the retinal features that we were interested in observing. In this case, cones that are right on the border of atrophy in comparison with cones that are clearly within that what's considered to be the retained central island. You certainly could adjust the size here. We wanted to go small enough that we'd be able to have this transition. We also have to keep in mind the normative structure of the visual system. And so these borders in choroideremia are located at various eccentricities relative to the fovea. And individual cones may not, in normal conditions, as you move further from the fovea, a single cone may not provide a full visual stimulus all the way up to cortex where a person would recognize and say, yes, I saw something, because of neural pooling. And so that's the reason for not picking a stimulus that's single cone sized, because we were looking at eccentricities that are outside of that foveal region.
And what method did you use to find the threshold, like to check the sensitivity? And actually,
Sure.
How bright is the brightest stimulus, or how dim is the dimmest stimulus in your system?
Right. So the units here are in decibels, and it really is a mechanism of contrast. So there's always a background intensity that's presented to the patient because we're imaging. And so you have to keep in mind essentially the background light in comparison to then how much contrast can you bring out in your stimulus presentation.
Okay, so if you're imaging, you're using what exactly?
It's a
more or less
795 nanometer beam. It is visible, so I know it's in the near infrared, but people do perceive it as kind of a deep red. So you do have that, you always have to keep that in mind. Here we haven't, the spot that's marked by the red really was the brightest that were able to deliver the light through the system.
And the stimulus is white or other color?
The stimulus was a 550 nanometer light.
Okay, and
So
the
yeah,
color?
agreed.
Okay. And what kind of strategy did you use to find this?
Sure. So the protocol uses what's called a quest staircase. Okay. So it starts at one particular contrast level, and then either depending on the person's answer makes either a large increment change higher or lower contrast. And then as each trial comes, that step size becomes smaller. The goal being that by the end of the Quest staircase, you've arrived at your threshold sensitivity, which is defined as a certain percentage of seeing. I think our percentage was around 75%. I'd have to go back and look at the exact paper methods, but that's on the order of what it was. So like 75% threshold seeing. So you do this stepped Quest staircase to get there. I think we used 20 trials per staircase and ran each location at least 3 times.
Thank you so much.
Of course. All right, so using those same methods now, here's an example of our retinal at an outer retinal tubulation. Again, we've now tested that Quest staircase protocol at these different eccentricities these different locations, all close to the outer retinal tubulation, which is coming off on the left side of the image here. What you find again is within that centrally retained island, we have measurable thresholds. These cones are contributing to measurable perception of the stimuli. There's the transition zone, but this transition zone actually kind of occurs as if you were to just continue the border, sort of lopping off that outer retinal tubulation. And so the stimuli that are presented over that outer retinal tubulation are again not contributing, or we're not able to measure a threshold there to the particular stimuli that we used. And so these results were published a couple of years ago now in Ophthalmology Retina. So I really enjoy the adaptive optics microperimetry. It's fun to me to be both the testee and the tester. I've sat on both sides of that experimental protocol. But in all honesty, it's tedious, it's slow, it requires a lot of concentration, and the required methods for zeroing in on a threshold at each of those small spots, even within a small region of the retina, really means that it's not going to be a high throughput method for either a large number of spots or a large number of patients. You really need to direct your experimental design to make sure that you're testing to answer very specific questions about the condition that you're looking at. So we've been interested in trying to understand whether we can identify biomarkers within our images that would allow testing of function of cells in mass with a much shorter and easier protocol for data collection. And so this enters now into the field of what's called optoretinography. So optoretinography is a technique for measuring an optical signal that arises from the retina in response to a visible stimulus. And the corollary really is electroretinography, or the ERG, which has been known for a long time to allow investigators to measure an electrical signal that comes in response to a visible stimulus. And optoretinography in and of itself is not a new idea. In fact, the first publication of it was introduced in 1994. Although at that time, the investigators were unable to find an optical signal. And I think really the results that are coming out now have arisen from our imaging capabilities, and in particular, the adaptive optics, being able to see, visualize, and track individual cells in the retina and pull these optical responses from the cells themselves. And so in recent years now, there's been, what's becoming a true body of work to develop optoretinography techniques. So what does this signal look like? In my group's hands, we're using adaptive optics SLL. So we have an on-phos view of the retina that shows the photoreceptor mosaic. And so here's the types of signals that we are able to get out. I truly hope that this video is playing well over Zoom. It is high resolution on my screen, and I hope it's showing efficiently on yours. But so we image the retina using our infrared imaging beam, and stabilize the video through our post-processing registration techniques. But while we're imaging, we also provide a visible stimulus that goes over all of the cells. It includes the entire imaging field. And then we record the video before, during, and after that visual stimulus. And once that is done in post-processing, we're able to track the reflectance of individual cells that occurs in response to that light. So here's an example of the cone outlined in orange. I've just enlarged it over here for you to see. And then below that, we're plotting the cell's reflectance as a function of time. And so for this cell, it starts with an initial reflectance. Our visual stimulus comes on. That's noted by the red bar, the red flash in this video. And when that happens for this cone, the cone signal increases and then decreases its reflectance with response to that stimuli. If we look at a second photoreceptor, this cone again starts with an initial reflectance. The stimulus turns on, and then this cone's reflectance decreases with response to the light. And if I then look at a third cone, this cone also shows a unique trace. It increases and then decreases and then increases again, or oscillates, but still in response to that light. And so what I'll impress upon you is that we made this observation that the cell reflectance is highly heterogeneous, but that the photoreceptors, that change in reflectance is happening with the presentation of the visual stimuli. So one of the questions that my team set out to answer is, to what extent can we use this change in near-infrared reflectance as an optical biomarker of cone function? And we spent a long time trying to understand how best to quantify this signal. If we finally settled on a technique that standardizes the cone reflectance using the pre-stimulus conditions, and then looked or measured essentially the change in the cone reflectance following that stimuli. So here's a video now where the cone reflectance is standardized in the pre-stimulus condition. The stimulus comes on, and you can see how the photoreceptors really wildly vary their reflectance from that initial starting value. But then plot each cone's reflectance as a function of time. So each individual red trace in this graph that's come up is a single cone's reflectance signal. And in that pre-stimulus condition, you see that they all start with that standardized reflectance. And then when the light turns on, noted by this dotted line, the cone reflectances vary from that initial value. And so then we just summarize that by aggregating over all of the cones. And this gives us what now becomes essentially our optoretinography signal. We do subtract out a control non-stimulated condition, and that leads to what is our optoretinogram, or the ORG. So this graph here is the summary graph of that optoretinography signal. Again, before the stimulus, cones have an initial value, and then when the stimulus comes on, we see this peak in their ORG signal. Now we want to know, well, okay, to what extent can this be used to understand cone function? And so does this follow known properties of the visual system? The first set of experiments that we did is essentially a dose response, looking at if we change the intensity of the stimuli presented, do we get a change in the optoretinography signal? And the answer there is yes. So here's 4 different stimuli intensities delivered to the retina over 3 log units. And you can see that the signal increases as our stimuli get brighter. We then plotted essentially just the amplitude of the response. And that's what I'll be showing for the remainder of the section of the talk, is the amplitude of these curves. And so again, as we increase our stimuli, our stimulus irradiance, we get an increasing amplitude of the ORG. We undertook another experiment to look at the wavelength of stimuli presented. So we changed the wavelength of our stimulus and essentially measured the action spectrum of our optoretinography signal. And this is shown here in the orange dots from aggregated over now multiple subjects, essentially showing that the action spectrum of the optoretinography signal matches that of the human photopic luminosity function, really providing the evidence that this signal is related to what we know is a functional response or functional vision. And this was our first optoretinography paper published in 2017 by our group. So I have a postdoc who's been interested in trying to take this signal and turn it into a biomarker that we can use to assess cone function, both in health and disease. There's a number of experiments that we need to do in order to understand and be able to apply this technique to our study of disease retina. And so the first thing is, like, is this a repeatable measure? So my postdoc took images and took a set of data with a particular stimulus, with both within a session and in a second session. And then he repeated that same measurement. And so plotted here is the first part, or like one group of that particular set of stimuli and optoretinography signals, along with the peak amplitude. And then when it was repeated within the session, we essentially get the same response, showing that our optoretinography amplitude is repeatable within a session. He did a number of different experimental conditions, different stimuli conditions within that first session. And then we actually had everybody come back a week later, and we repeated the exact same imaging protocol. And so this now is comparing session 1 to session 2, again, showing that our amplitudes were repeatable within sessions on both times, and then also were repeatable across sessions. And if I just summarize that over a number of subjects, here I've plotted now our session 2 amplitudes on the y-axis in comparison to the session 1 amplitudes on the x. And we found that there's no significant difference in the amplitude within the session, no difference between the sessions. But interestingly, that our normative subjects had a range of ORG responses with significant differences between subjects, showing that we'll have individual variability across the normative population. And this really provides motivation for doing studies that will identify population norms so that we can really compare to different disease conditions. What I've shown you so far related to the optoretinography is looking at responses where we've pooled across cells. So we've really harnessed the power of the heterogeneity in the response from multiple cells within the mosaic. And we'd really like to get down now to single cone function. Can we come up or use a technique that would allow us to get a single cone ORG? And the response there, or the answer there, is yes. We've not now, instead of pulling across cones to get a population ORG, we've actually been able to pool across trials. And so we do multiple flashes and pull out a cone's reflectance trace. We find that that reflectance trace is also heterogeneous across trials. So we can use the same process of pulling multiple trials for a cone in order to get an individual cone's signal. And this now allows us to essentially give a functional response or functional marker to each cone within the mosaic, individual cone ORG. And this work was first published from our group in December of 2020. So now that we have this capability, we can proceed or start to think about how do we use it in patients. So I'm going to bring us back now to our choroideremia subjects, where we've looked at this optoretinography response in a group of choroideremia compared to a group of normal-sided controls. And what you see is, like, the cone mosaic in the choroideremia subjects, they have close to normal cone density in this subgroup, probably a little bit reduced, but near normal for sure. But when we look at their optoretinography response, we find a significant difference in the amplitude of the ORG that our CHM subjects exhibit in comparison to controls. And so this is definitely something that we're looking forward to continuing to investigate in order to yield a biomarker of cone function within these diseased populations. So now I want to transition and talk about how we can use these methods to understand not only what's going on in the natural occurrence of disease, but also what happens when we intervene. And, as ophthalmologists want not just to understand their patients and what's going on with the patient's vision, but also to halt degeneration or in the best case, even restore visual function. And so I've been incredibly, it's been an incredible experience to be able to work with so many wonderful colleagues at Penn and around the world that are developing treatments, novel treatments for inherited retinal degenerations. And it's an exciting time in this field because it's only really recently that we've been able to think about or talk to patients about the possibility of having treatments in the future for these degenerations. So there's ongoing clinical trials for choroideremia, and one of those trials did occur at Penn. So my group was able to recruit the same subjects that underwent gene therapy for choroideremia and image them both before and after. And so let me just talk for a minute about gene therapy or the gene augmentation process. So as I said, there are clinical trials in process. The idea is that you will deliver a normal copy of the choroideremia gene to the retina via what's called a subretinal injection. So through a surgical process, there is a bleb created that essentially forcefully detaches the neural retina from the RPE. And this creates a subretinal space. And within that space, the normal copy or a normal copy of the CHM gene is delivered. And the idea is that the cells that are adjacent to that space, being the RPE and the photoreceptors, ideally both, but that they will take up the normal copy of that gene and therefore will be able to produce the Rab escort protein 1 that they're lacking. And so that's the idea behind it. And so, my team said, well, that treatment is occurring at the cellular level. We have the tools now to see the individual cells. And so what's happening as the photoreceptor mosaic before in comparison to after this subretinal injection. So we were able to do that. We did follow all 9 patients. This is an example of one of the subjects before and after, where I've taken the two adaptive optics montages and overlined them. And in a, just qualitative appearance, you can see a lot of the same features from one time point show up in the later time point. For instance, these locations marked by the yellow arrow show the border of atrophy or the photoreceptor mosaic within that retained islands, and even just the more macroscopic views of the retina that we can track over time. But we really are interested at the single cell level. And so here's now a zoom in of just one region of that mosaic, where I've placed boxes on this image here to help direct where you might look or see a group of cells in one image that is maintained in the other. And so we were able to do this with all of the subjects, and we took regions of interest now, both in the baseline or pre-injection time point in comparison to 1 month after the gene therapy injection, at regions of interest both in the treated and the non-treated eye. And then from these, we were again able to quantify the mosaic using metrics similar to what I presented earlier in the talk. And so here are cone density measurements made from this one particular subject's regions of interest before and after. So if I look at the if I look at the cone densities plotted 1 month post-injection in comparison to pre-injection, this is for all 9 participants, but excludes the foveal center in one person for right now. So it has the green dots are from the injected eye, and the gray triangles are control eye. And what you see is that when you look at cone densities before and after, they are really the same between those two time points. And indeed, we found there was no statistical difference in the cone densities of these regions between time points or between the injected or uninjected eyes. And so these results lead to 2 important safety conclusions related to the gene therapy. So first, the surgical intervention itself does not cause widespread cone loss. You can induce this retinal detachment, the photoreceptors resettle, and by and large, they are maintained in the short term following that intervention. And then the second important safety conclusion is that the study agent did not cause an acute toxicity, meaning that 1 month time point, the photoreceptors are still maintained within that mosaic. And this was work that we just published in the spring of this past year. So I do want to take a minute though, because there was a 9th participant right in the foveal center that had a little bit of a different result, which I do need to mention. So here is this mosaic of the central foveal region in that 9th person. The areas in the yellow, outlined in the yellow boxes, are kind of the very close parafoveal regions. And if I zoom in on those at that 1 month post time point, you do see the photoreceptor mosaic maintained at those locations. But if I do look in the very center, this person does have a couple of spots here where the photoreceptor mosaic is no longer visible. And in fact, this person at the 1 month time point did exhibit a 12 decibel loss in cone sensitivity. So this raises the question about whether there's individual vulnerability at specific retinal locations or the foveal region in particular of these patients. And it's something to keep in mind as we proceed with gene therapy clinical trials, not just with choroideremia, but for all of the inherited retinal degenerations that are going into clinical trials. This is something that we want to continue to investigate and pay attention to. And again, this work was included in that same paper from last year. So with that, I will just summarize. We've talked a lot about the structure and function, particularly related to choroideremia. The cones were less dense and larger in choroideremia than control. We showed very tight correlation between structure and function, particularly at that atrophic border, with reduced optoretinography amplitudes within the central island. And our gene therapy assessments in the short term show that the mosaic is structurally retained following that subretinal gene therapy, with the one asterisk about the individual vulnerability of our single subject at the fovea. And with that, I'll just end by saying, adaptive optics ophthalmoscopy is a tool now that we can use to investigate these the structure and function of the visual system in these different conditions. So with that, I'll thank you for your attention. I'm more than happy to take questions both now or also after, if you want to reach out by email or Twitter, that's great. Thank you so much.
Tremendous progress, wonderful presentation. I have a quick question to you related to the cone responses. As you have shown, they peak somewhere around 1 second or even longer than that. That is not compatible with electrophysiology of cones, which are much, much faster, right?
Yes, great question. Yeah, so the stimuli that we were using that I showed here is they're definitely longer than typical flashes. We're using, for most of the work I showed, it was a 2-second stimulus. We have started to go faster for that, but you're right, the time course of the response is much longer. From the OCT world, we are starting to understand the mechanism for what's causing this response. And it really does seem to be caused by the multiple reflections that arise from cones. Depending on which set of nomenclature you use, that's either the inner segment, outer segment junction, cone inner segment tip, or in the nomenclature of elliptical zone and interdigitation zone. Those two reflectance layers of the retina in SLO or adaptive optics SLO appear together. And so essentially a cone can act as a little interferometer with these two reflections as they change in their distance or to each other, or really the optical path length between those two. That can result in either constructive or destructive interference. And so we really do think from the OCT community that it is a lengthening of the outer segment, very, very small, on the order of nanometers, but a lengthening of that outer segment that contributes to this optoretinography signal.
Right, let's get to other questions. Final comments will come from Professor Wojtkowski. And the first question goes to Kamal and Bruce afterward.
Oh, okay. Hi, Jessica. Beautiful talk.
Thank you.
So it seems like you're taking a coronal section through a cone, right?
Mm-hmm.
And measuring like the max reflectance from that two-dimensional slice. But the shape of the reflectance profile along the axis of the cone, as I've shown and others have shown, will affect the reflectance measurement. So if you investigated the cone shape profile, not so much the lengthening, but the actual profile of the shape.
Great question. So no, we have not looked at how that shape might change. We've really just taken a projection through the photoreceptor, and that's certainly somewhere we could consider expanding the results. Rather than take just a single pixel, though, we are aggregating over the pixels contributing to the shape. So it is kind of an average measure of the reflectance over that cell's expanse.
I would just say you don't walk across a stream that's got an average depth of 2 inches, right?
Yep, absolutely.
Thank you.
Let's move to Eric because he's anxious with his hand.
Oh, I didn't know whether you could see. Hi, Jessica. That's Eric Hartman with the Choroideremia Research Foundation.
Good to see you, Eric.
Hey, when you're talking about the use of this as potential endpoints, I know that advanced CHM patients can have problems fixating in order to be still enough for these individual measurements of cones. Would that become a stipulation if you're talking about using these as endpoints, that the patients are going to obviously need to be able to keep fixation?
Sure. So thanks for the question. It is definitely easiest for our image collection when people can fixate and eye motion is minimized. I can tell you not just from work with choroideremia, but in other inherited retinal degenerations where the eye motion is not as steady, we're still able to get results like this. So we have had people, even with mild nystagmus, be able to undergo these experiments and have obtained successful results. So that, I think, is, as our technology improves and continues to improve, in particular with eye tracking capabilities and things like that, I become less and less concerned for whether, you know, in the long term, we'll be able to really undertake challenging conditions. Let me clarify something, though, related to outcome measures. So during the part that I was talking about, that was in reference to the microperimetry results, which showed very close correlation between structure and function. And so I love adaptive optics, SLO. That's, you know, my bread and butter from a research standpoint. But from a clinical trial endpoint standpoint, OCT that shows the extent of the photoreceptor layers that remain intact would be perfectly acceptable. That's a good structural marker. It doesn't have the same resolution as seeing individual cells, but you know, understanding the spatial extent that we have those layers intact could easily fill the marker for a structural endpoint.
All right.
I'm sorry, just a quick follow-up. So we'd need to have a study that could correlate that to then prove to Sieber and the other reviewers?
Yeah,
that
I know
endpoint
it's
would be
always tough
viable?
because you got to get through the FDA, right?
Right.
And I would argue that our results already show the structure function correlation.
Okay, thank you.
And yeah.
All right, we have a comment.
Hello, Jessica. This is a Kamal Ragal here in FB. I don't know whether you know me or not. I joined just after you left David William Labs, right? 2017. That time. Okay. The one question, you know, that your result on optoretinography is really exciting, but you showed there is no difference between the two sessions on the amplitude. But do you have any data for the longitudinal study of the one patient? For example, if we like to study the drug effect, what happens over the time, the reflectance, do you think would be a, you know, like, as I remember, the cone has the orb guide property, right, which changes where it sees the eye. So if the person look at different time, then your amplitude might be different with the different time point, right?
Absolutely. Yeah. So that's exactly what we want to do. And in fact, we want to do that. I mean, when I think about future applications for this, I can imagine longitudinal time points before and after therapy, not just trying to see if there's a difference, but also seeing if there's a difference on single cells. And we suspect, I think from preclinical work, that not 100% of the cells won't take up the therapy. And so I actually think about this from a standpoint of, like, could we determine which cells have taken up the therapy as opposed to which cells haven't? And could you essentially make a picture of which cells you think might retain function in the long term? So that's a dream of mine. I would love to do it. And we're actively working on making that possible. We don't have the results to show from the 1 month time point data that we showed right here. And in part, that's just a matter of how the fields have arisen. So, when the clinical trial started, optoretinography didn't exist in the form that it does today. So it's definitely a project moving forward.
But do you think, like, that the reflectance from the cone itself changes depends upon the where the eye is oriented, right? Wouldn't that interfere on your result if you continue, or it will be average out?
I see what you're asking. Okay, so there certainly are studies that show the initial starting point of the cone reflectance changes over minutes, hours, days, and depends on pupil position. We certainly know that. What is nice about the method we're using currently is that we have this standardized, we're really looking at a change in reflectance. And so our measure is essentially irrespective of the starting value.
I see. Okay, thank you.
Let's move to Arthur, and after Arthur, Grażyna.
Hi, Jessica. Thank you for that wonderful presentation, fascinating results.
Thanks, Zach.
I wanted to follow up on your on Chris's question and your discussion about the peak being slow, but also it appeared the tail seemed to be quite slow. If I remember correctly, it was still quite high on some of the responses you showed at 5 seconds. So that, do you expect such a kind of a slow recovery from a physiological point of view?
Yeah, that's a great question. So I'm definitely speculating here because we have not done any experiments related to time course. But I think that part of the tail that we might see really is a result of it could be in part a result of how our measurement is made, which involves under a comparison to the cell's initial reflectance. And so we kind of think that the initial optical path length between these two reflective surfaces is kind of like, there's the optical path length, let's say is 30 microns or something like that. But each individual photoreceptor be plus or minus a little bit. And so the exact phase is kind of randomized around that, average value, average length. You with me so far? Okay. Yeah. So, so then you hit with the stimulus, the cell or optical path length, maybe, well, from OCT, we believe that there's a very short shortening that occurs quickly. And then there's a longer lengthening that occurs. And where it ends or where it comes back to is going to be, again, randomized from cell to cell within that, 30 microns plus minus. So what we have not done yet is really experiments to understand, if the cell resettles at a slightly different spot than what it started, is that going to result in us seeing this kind of long tail? And there I'm not really sure, but I think that might have something to do with it. The OCT community has shown these, similar long time course results as well. Their result is also not anywhere close to the speed of an ERG. And so I think there's just still a lot more to understand about this. I have not seen publications from the OCT looking at, what happens after the lengthening and does it return. I don't think that there's kind of seconds long of data that's been published there. So it's something I'm certainly interested in continuing to follow. Although you are doing multiple recordings from the same cell, so you must have an interstimulus interval that you are allowing for recovery to be able to do that. So are those long, are those short, or what? Yeah, so good question. In practice, right, we want to collect a lot of these responses, a lot of these flashes. So we I want to say our recording time at present is a couple seconds post stimulus. So we have the person blink, blink, look in, a second before the stimulus flash, a couple seconds after their stimulus, our recording ends right there. And then we say, okay, blink, blink, and then we're going to push the button to go again. So, that there's some variability in before and after. The thing is that every time we push record, there's still this pre-stimulus interval. And so every recording is standardized to that one pre-stimulus. We really haven't tried to put together like the full time course back to back. That becomes much more difficult because it's broken up by allowing the person to blink, which we have to do. We have to let them blink. So that's part of it. We do ask we try to collect about 10 in a row, and then we have the person sit back, take a 2-minute break. While that 2-minute break is happening, we have them patched so they, in theory, are getting kind of back to a steady state. In one of our publications, I think we did publish what we think would be the bleaching dynamics for the length of these multiple record in series recordings. So could direct your attention there.
Thank you very much.
Welcome.
Right. So I was going to ask something similar what Arthur has already asked, so I slightly modify. So the size of the cone that you see on the SLO is basically the size of the inner segment. However, you make the argument that the signal that you get is like the compilation of the signals that you get from the tip of the outer segment, if I correct, and also from the intersection between the inner segment and outer segment. So it's hard to say exactly what we are looking for. The OCT tells us that the optical path is increasing, but also there was some motivation and logic that said that this is related to the osmotic pressure in the outer segment. So this is extremely interesting question. Do you have any inclination how is it possible to tease out whether the signal is really from the outer segment that you see in the reflection, or maybe this is because we have all this NADPH in the inner segment? Do you see a way of differentiating using the SLO?
Right. These are great questions and experiments that I would really like to do. I kind of glossed over a little bit where the images arise from, and from a technical standpoint, we can actually collect what we consider to be multimodal images all from the same device simultaneously. So the optoretinography work is done using what we call confocal reflectance. So we're really getting the signal that, similar to a confocal microscope, the signals that are, directly focused back out related to that specific area in the retina. Now, from our resolution, our axial resolution in confocal AOSLO is nowhere close to where it is in OCT. And so our axial resolution, we do believe, will encompass both of those reflectances at the same time. And so you can't really tease it out in the confocal signal. Separate to that, we also have the capability of using what's called non-confocal split detection. And those images are collected under what we consider, they arise from what we consider to be multiple reflections or multi-reflections within the retina, kind of scattered. And the size measurements that we made of the inner segments arise from those non-confocal split detection images. And we really do believe, in particular from what's been published in the literature, that is a measurement of the outer segment extent. So the size experiments that we did were related to using those split detection images. Now, whether we could get an optoretinography signal in our split detection measure, which we think would come from the inner segments, is still yet to be determined. But it was one of my proposed experiments to do in the R01. So pending, I guess.
Thank you.
All right, let's have a last 3 questions from the room, and then we move to Professor Wojtkowski to summarize all. So Sam, Rafał, and Vladimir.
Hi, Dr. Morgan. Great talk. My name is Sam, and I'm a grad student in Chris's lab. 2 quick questions. Have you ever tried giving 2 really rapid successive stimuli in the ORG? Like, for instance, when the first ORG signal peaks, what happens if you give another stimulus?
So that's a great question. No, I have not done that experiment in the exact way you just described. But what we do know is that the signal will saturate. So there's a limit to how there's a limit to how bright we can continue to make that flash and continue to measure an increasing signal. Probably from a physiological standpoint, this makes sense. If the signal is arising from a lengthening of the outer segment, there's going to be some limit to what that outer segment can expand in a short period of time. So I think from our dose response measures, if you stimulated multiple flashes, you would expect that stimulus to continue, or I'm sorry, you would expect the signal to continue to grow up until you reach the part that you would be saturated.
And have you seen a response in rod photoreceptors?
Great question. Yeah, we would expect the signal to be also in rods. We haven't measured it ourselves. There is a publication suggesting that the rod photoreceptors also show this type of response.
Hi, Dr. Morgan. I'm Rafał. I'm a postdoc in Chris Polczewski lab. I have 2 quick questions. First, why do the cones in diseased retina appear larger? And second, if it would be feasible to deliver the AAV to choroid to kind of treat the choroideremia upstream where it really begins. Thank you.
Great. Okay, great questions. So yeah, this increase in cell size is something my graduate student is currently putting her ideas together for what is going on so that we can submit that work for publication. It's really fascinating to think about whether these cones are getting larger as a process of disease or just because they have more room and can therefore expand out. There are different, there have been different conditions that have shown, for instance, like if you damage some RPE cells, the neighboring RPE cells might expand to kind of like fill up that mosaic. And so that's one hypothesis, that these cones just have more room. And so they kind of like relax and get bigger and take up all the space, or take up, as much space as they can. So that's one idea. There's another idea that says, hey, this is these cones are sick and diseased, and their morphology is different. They're on a path to degeneration. And so, perhaps there's differences in, like this is one step of that process. And I'm not sure that our results right now can fully differentiate between those 2 possibilities. We're interested for sure in doing longitudinal studies to see, if we can tease out more information from following up these same photoreceptor cells over time. And related to your second question then was related to treating the choroid, and that so it's an interesting idea. I think that the studies looking at the mechanisms of degeneration in choroideremia have started to investigate what are our primary targets for treatment. We see the degeneration in the photoreceptors RPE and choroid, but it may turn out that if we rescue, for instance, the RPE, that that there's a question of, well, would the choroid still continue to degenerate, or by having the RPE, would the choroid be able to be retained? And I think there's literature to suggest that the choroid really is a secondary loss effect, although I'd have to go back and really dig into some of those mechanistic studies in the literature. I think the question about photoreceptors is still outstanding. We believe that the rods are a primary source of degeneration. These patients lose their night vision first as their first sign of what's going wrong. But whether the cones are degenerating as essentially a secondary mechanism to losing the underlying RPE and the surrounding rods, and eventually the cones succumb, or whether the cones are a primary source of degeneration, that even if you rescued the RPE, they would still continue to get lost, is still an outstanding question.
Thank you so much.
Hey, Jessica, beautiful, very exciting work and beautifully presented.
Thank you.
Following Bruce's comment on the size of the cones, of course, peripheral cones and foveal cones have different shapes. They also have different physiology. So my question to you is, have you compared your ORG responses from foveal and peripheral cones?
Fantastic question. That experiment is in progress. And actually, in particular for the students out there, my lab is taking the approach of pre-registering our experiments. So we lay out the experimental design and then actually post to the Open Science Framework website what experiment we're undertaking, not as a it's not as a strict contract for what we're going to do, but really as a signposting of this is what we've designed, and if we choose to deviate from it, then we know, and the rest of the world will know too, because it's out there and accessible. But anyway, so that's an experiment that we're currently undertaking. Yeah.
All right, let's move to Maciej. Maciej, all yours.
Okay, thank you very much, Chris. Thank you, Jessica. So that was really great talk, and I really appreciate these examples, how we can transfer the new technology to the clinical cases, and also, to show the potential that still with these new optical imaging methods, you can find out new ways of understanding the processes related to vision. So I'm very big fan of optoretinography too, and that's why we are still developing a new way of optoretinography called FORG, which is the flashing or flicker-based optoretinography, to focus on this short-term phenomena that is related to change of the photoreceptor thickness within milliseconds. And so with that, we believe that this is incredibly appealing to replace in future microperimetry and ERG. And for many ophthalmologists, even if they don't yet realize how huge help it will be with the monitoring and diagnosis. So the question is still, there are some concerns about full understanding of the physiological process responsible for the changing of the thickness of cones, and what causes really this condition from the standpoint of vision process. So that Jessica just demonstrated us in these beautiful examples. There are some rather speculative theories right now how to explain it, but this is still great challenge to many researchers, not only engineers, but also ophthalmologists, biochemists, vision scientists. So, and I also wonder, as a last remark, if you can change slightly your protocol to photopic conditions, and try to look at very small changes in phase in shorter time scales, to see if you can really follow this reaction that would correspond to the real process, in 15, 20, or 50 hertz time frame. So how do you think, is it really the experiment that you can conduct with your system?
That's a really interesting question. What I'll say is that my system right now is limited in speed by our frame rate. So we're operating in about 18 hertz, which is really the minimal measurement that we can make. So there is a limitation of speed there. I do think that we're probably operating right now in some type of mesopic state. We're not fully dark adapted, but we do have this, very brief, let's try and get the cones to some kind of steady state to begin with. So I think there are adjustments that we could do there to operate in a more photopic region. And I think we would still see the signal.
All right. Jessica, thank you very much. You had a lot of questions. I hope this was helpful to you as well.
Fantastic.
Thank you for your talk. Thank you.
Thank you.